Okay, so as the last part of muscle discussion on muscle or muscle labs, we're going to look at neuromuscular junction. So first off, I want to tell you, we're going to go very deeply into the muscle physiology, all the action potential and muscle contraction mechanisms very well in the lecture series. But I just want you to introduce to you neuromuscular junction because this is very important. And at the same time, I don't want to go very deep into the neuronal part of it either, because we are going to be looking at it in the next set of lab and lecture sections as well. So basically what we are trying to do at this part is the sort of like the first page of your exercise number 11. That's all. But then I want you to please look at the pictures that I'll be putting out in the lab, even though that is not in your book. Please understand how the muscle motor unit is, like you have what is the part of the muscle that actually causes the contraction and so on, right? And so that is not given in very good detail in your manual, but like I said, use the printouts and then also the slide that I'll be showing you in this one. Okay. Some of it may be repeated in your lecture session, but you need to understand. So basically you have a muscle, right? And let's say it's your gastrocnemius, right, at the back of your calf muscle, right? And so and then you have your quadriceps and so on. Yeah. So your thigh muscle and so on, right? And so let's say you are uh, you're walking and then suddenly the tiger is chasing you, right? And so then you got to run. That means your muscles have to get going. But how do they know that they have to function? They have to contract and relax and make your movement fast movement possible, right? So basically they have to be dictated by a signal that comes from your nervous system, right? And so your neurons, your I see the tiger, and they process the information, send it to the brain, and then immediately your brain sends a lot of information, right, via the axons and everything. And then so that electrical signal basically comes through the axon, right, maybe jumping through the nodes of Ranvier and everything, but then eventually it reaches the nerve terminal, right? And at that time the electrical signal is turned into a chemical signal, because now the chemical here is your neurotransmitter. It could be like acetylcholine or other kinds of things. Like you can have dopamine as a neurotransmitter, you can have glutamate as a neurotransmitter, and so on. So these neurotransmitters are going to be packaged into synaptic vesicles, right? And so a synapse is any joint, a region where a cell makes contact with another cell. So you have your neuronal cell in this case, right? And it basically says, okay, this is a muscle cell, I have to communicate with this muscle cell. So this area of around the neuronal ending, right, that is called your synaptic terminal, but it's also called presynapse because it's the direction of the signal. The signal comes from here to here. So this area is your presynapse. Then between your presynapse and your postsynapse is the gap, right? That is called your synaptic cleft, right? And so the postsynapse cell doesn't have to be always muscle. It can be a muscle, it can be another neuron, it can be something like a gland, right? Endocrine gland. So the nerve comes and says, okay, you got to secrete some hormone, like that. So here let's just say, but here we are going to only look at your muscle. So you have your nerve in the presynapse and your muscle cell in the postsynapse. Therefore this junction altogether is called the neuromuscular junction, or NMJ. Okay, so basically what happens is, like I said, this usually is depicted as a conical sort of a area, and then you have the axon, right? So from the cell body of the neuron, the electrical signal comes through the axon and then it reaches the synaptic terminal. And at that time, when the electrical signal comes there, then there is an increased influx of calcium into the synaptic terminal, which basically tells the vesicles with the neurotransmitter, go fuse with the plasma membrane. So that's your plasma membrane at the synapse. So this area where the synaptic transmission happens, there you have varicosities of the membrane, presynaptic membrane. Those are also called synaptic varicosities, some people call it synaptic boutons, right? And so these are little swellings. So at those parts, the vesicles are going to just fuse with the plasma membrane, like that, exocytose, and then the neurotransmitter is going to come into the synaptic cleft. Okay, now at the other side of the synaptic cleft is your muscle cell, right? So the membrane of the muscle cell, that is your post-synaptic membrane, and that has many receptors specific to the neurotransmitter. So now they'll endocytose the neurotransmitter, right? And so now you had the electrical signal turned into chemical signal, and now this is going to go back and then start electrical signal, because it will allow calcium to enter. And then, okay, so now calcium enters the postsynaptic cell. When calcium enters the postsynaptic cell, it can cause it will cause muscle contraction. So in skeletal muscle, you will see increased contraction of the muscle proteins. They align in such a way, they interact, and then so the muscle starts to contract, and now it's an electrical signal. So in smooth muscle, you can also have calcium influx, but increased, because the endoplasmic reticulum or the sarcoplasmic reticulum will release the store of calcium, and that will do its thing. So you can have different effects because of all this contraction signals and so on. Okay, so now what happens, what is the assembly? This is why muscle is so beautiful, right? So like I said, the nerve we saw is a cell body. You have the axon and the synaptic terminal and everything. In the muscle, what actually happens is you can form what is called a motor unit. That is where a motor unit is where one neuron or multiple neurons basically contact a bundle of muscle, and that signal comes from that neuron, and that muscle responds, right? So together this assembly is called a motor unit. But anyway, in the case of a muscle, generally when we look at your arm muscle or whatever muscle, you have a muscle, right? So that is a muscle fiber, and a muscle fiber can have it is made up of uh, myofibrils, right? Or sorry, the muscle, individual muscle is bundle is made of many myofibrils, right? And they have many discrete units called sarcomeres. Now the sarcomeres, I'm going to show that in a slide, the whole dissection of this. I mean, not the dissection as such in the virtual dissection way, but like the anatomy of the muscle fiber in just 1 minute. And then, so the sarcomeres are going to be attached to each other, and that line where they attach, the boundary line, that's called your Z lines, like Z, right? And now these myofilaments that are very beautifully arranged and very beautifully arrayed, and that is the reason the Z lines are aligned and everything. That is the reason the striated muscles have the striated appearance. Now there are other muscle specific proteins, like you have a muscle specific actin, you have myosin, you have tropomyosin, and those proteins are going to be involved in the muscle contraction. Okay, so in the muscle, typically the nerve, when at the neuromuscular junction, when the signal comes from the nerve, the part of the muscle that is receiving the signal is called transverse tubules or T tubules, right? And because of that, when that receives the electric signal, then calcium is released from the sarcoplasmic reticulum, like I said. That is an organelle inside the muscle, and now the actin and myosin are going to be interacting, and that will cause the power stroke or the contraction of the muscle. Okay, and so you will also learn later in muscle physiology a principle of depolarization. So generally the muscle membrane is going to have a negative charge, right? But then when this happens, when it is going to respond to the neuronal signal, then the negative charge outside and the positive charge inside. So it's it's polarized. So you have two different charges across the membrane. But then because of the response to the signal, then suddenly you're going to lose the negative charge. And so the charge is going to become positive. And that means you're basically losing the polarity between across the membrane, right? That is called depolarization. So the charge is going to go up toward the positive side of it, and then slowly it will come back, and that is called a repolarization, right? And so every time you have the polarized status to begin with, then you have so that will be negative to with respect to 0, and then from the negative it will go depolarized, and then it will get repolarized, right? So that is called an action potential. And so that is going to happen according to the frequency of the action potential, according to the amount of calcium release, and how the polarity is re-established, and so on. The polarity, by the way, is established because of ions. That's why it's an electrical signal. So you have positive ions like, sodium or calcium, then that area is going to become a positively charged area. Whereas if you have something, an area which doesn't have as many, that is going to be relatively negative. Also, if it has chloride or some kind of negative ions, it's again going to be negative. So the inside of the membrane versus the outside of the membrane, you look at the number of or amount of charges, and then you determine which is negative, which is positive. That's how polarity is established. Okay. Anyway, so we have the signal, and then, like I said, calcium is released from sarcoplasmic reticulum, and so the actin and myosin like sort of move across past each other, right? It's called that is called the sliding filament model, and then it will show that to you in a minute. And like I said, the muscle physiology will go in deeper during your actual physiology lectures. Okay, so now I'm going to show you the sliding NMJ first, neuromuscular junction.
An action potential arises at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Okay, so sliding filament. So this is what I meant, right? So you can see the Z band. So this is basically the Z disc to Z disc, right? So this is your sarcomere. It's actually showing here. So the everything that is your unit of a contraction, right? So you have many of these sarcomeres in each myofibril, right? And then the fibril becomes your basically your myofilament, and then you have your big muscle together. So now in the sarcomere, you have these Z bands, like I said, and so because of this, right, you can actually see how they are arranged very precisely, but alternating like that, right? And so that is basically causing the striation. Okay, let's look at.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. the actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
Okay, so yeah, so you just want to remember that. So this is your actin and that's your myosin, and so they are sliding, back and forth and so on, and that's how the contraction occurs. Okay. So let me give one example of your, say, your trapezius muscle. Oh, no. So that's just your muscle movement, right? Uh, or your pectoralis major. So like that. Yeah. So now what I'm going to do is the last bit of your thing. And so this is the last part of your muscle, but we are going to go back to chapter 10. You remember we left off the last bit of the chapter 10, actions and joints at joints. So basically here, hopefully, because you have now finished your skeletal system and your muscle system, so you kind of understand how their interaction at the joint is possible, right? And so basically you're going to we looked at the joints. I hope you remember that part of the skeletal system. Then the actions in the joints, I just want you to go over those paragraphs starting from 145, page 145 through 148. So there mainly it's pictures, right? So it also tells you the kind of movements that is occurring at the joints. For example, you have flexion, right? Flexion is, let's see, flexion is the decrease of joint angle from, okay. So, yeah. So, you have your, if you have a whatever a part of your body, if you decrease its length by kind of, you're flexing your muscle, right? So basically you decrease the length of that portion, that movement is flexion. By contrast, if you extend it, basically increase the length, you call it extension. So extension and flexion usually are opposite to each other, like that, right? Then you can actually have hyperextension, right? Sometimes it's very bad because your ligament can give out, and so, but basically that is overstretching. And then I talked about abduction and adduction in your lab class earlier on, right? And so that is where abducting means you're removing a bone or part of your body away from your body, right? So if you take your hip and move it like that, that's your abduction away. But then if you bring it closer to your body, that is adduction, like that. Then you have a rotation. You can have your medial rotation and lateral rotation. Circumduction is when you do this way, right? So basically it will form a conical. So this is the tapering end, and that is your big circular area, right? So that conical motion is called circumduction. You can do that with your leg also. You can put your leg there and then like sort of do that. Supination is lateral rotation of the hand, right? Um, and pronation is medial, right? And so on. So protraction is like putting your chin forward, protraction, right? And then retraction is like bringing it toward you. And then you have elevation, depression, inversion, and all this. So all of those movements are listed in your manual, like I said, page 145. And then in 146 and 147, all the movements are actually clearly indicated, right? So extension and deflection and, um, you know, yeah. and then you have abduction and adduction, right? So abduction is moving away, adduction is bringing it closer to your body, and so on, right? So if you move your hand like that, that's your medial rotation, right? So you sideways like rotation, and then that's basically your pronation, and then lateral rotation, right? So when you move it this way, that is your supination, and so on. Circumduction, all of those are there. So please make sure you understand these movements, right? And I think that is the end of it. I have spoken about 12 and 13. I finished earlier on. 14 we just finished. 15 we just finished. Okay. That's basically what is going to be included in your exam. And I will stop here.